Let's go now over PGY2320 Makeup Test 1 2023. And the question number 1 here, essential fatty acids. Now we know that essential fatty acids, these are fatty acids that cannot be made in the human body. They have to be gotten from the food that we eat. Can be made in the body. This is not true. Refer to fatty acids that are involved in metabolic reaction. Really not. Can cause cardiovascular disease. What causes cardiovascular disease? These are saturated fatty acids. Can only be obtained through the diet. This is the correct option. Number 2, using the Anderson-Hasselbalch equation, solve the equation. Solve the following question. What is the pH of a buffer solution made from 0.20 molar HA and 0.6 molar of A minus? pKa for HA is 5.5. Now we know that the generic form of an acid is given as HA. When it disassociates, it gives us a proton and also give us A minus. According to the Anderson-Hasselbalch equation, pH is equal to pKa plus log of the conjugate as base, which is the product here, divided by the acid. These are concentrations. So the pKa we've been given, that is 5.5, and then plus log the concentration of the conjugate base, that is a 0.6, or divide by the concentration of the acid, which is 0.2. So when we get to punch this, we're going to have 5.5 plus log of 3, because 0.6 over 0.2 is 3. 5.5 plus, so that is going to give us a 5.97, which is a 6. So that is going to be the answer there. What is entropy? Now, if you look at a system, say this is a semi-permeable membrane. Here you have got particles, and then you don't have particles the other side. These particles want to move in this direction. So that is what we refer to entropy, the degree of disorderness or randomness of a system. An expression of randomness of a system, because things want always to be random. They don't want to be packed together like that. Okay, so that is what we call entropy, the expression of randomness or the degree of disorder. For an enzyme that follows simple Michaelis-Menten kinetics, what is the value of Vmax if V initial is 1? So V initial we've been given, that is 1. And then we've been told to find Vmax, which we don't know. But we've been told that per minute at 0.1 km. That at 0.1 km, that is the concentration of the substrate. The concentration of the substrate is 0.1 of km. Km is the concentration of the substrate at which half of the active sites of the enzyme have been occupied. So the Michaelis-Menten equation, V initial is equal to V max multiplied by concentration of the substrate divided by the concentration of the substrate plus Km. So this will give us V max is 1. V initial is 1. V max is what we want. Concentration of the substrate, that is 0.1 of Km divided by 0.1 Km plus Km. So this is going to give us 1 being equal to V max 0.1 Km. 0.1 Km plus Km, that will be 1.1 Km. So here the Km cancel the Km. So remaining 0.1 over 1.1. So this will give us 1 is equal to 0.090909, just like that, Vmax. If you divide both sides by 0.09, we are going to end up with Vmax being equal to 11. So this is going to be it. A patient suffers myocardial infarction. Which of the following serum enzymes will be in higher than normal amounts? So when we are talking about myocardial infarction, and then you want to know the enzymes in high amounts, this one you just need to know there are 3 enzymes: aspartate, aminotransferase, creatine, and lactate. So you can use the mnemonic lactate, dehydrogenase, aspartate, aminotransferase, and creatine kinase. So this is the one here. Okay. So sometimes you've seen the way they put this one, alanine aminotransferase. So it's supposed to be aspartate aminotransferase. Sugar table is made up of glucose, and so sugar table is sucrose, is made up of glucose and fructose. Which of the following HIV genes encode for the HIV envelope protein? Right, for the envelope protein is supposed to be ENV. Envelope protein is ENV. The statement fatty acids are highly reduced implies that. What does the statement fatty acids are highly reduced mean? Highly reduced means that they have got a lot of hydrogen. That's the meaning. So for option A, fatty acids easily accept hydrogen atom. It's not about accepting. Fatty acids cannot get oxidized. Really not. Fatty acids have many CH bonds. This is true. Fatty acids have got free rotation. Really not. So about highly reduced, it is about the hydrogen. A lot of hydrogens. Next question. With regard to hydrogen bond formation, which of the following statements are true? Hydrogen bonds are formed between water molecules and the other polar as well as non-polar molecules. Hydrogen bonds can be formed between among or between polar and non-polar, so that is not true. The positive charged hydrogen atoms form strong interactions with more electronegative atoms. This is true. For example, it can form hydrogen bonds with things like nitrogen, oxygen, and fluorine. Carbon atoms participate in strong hydrogen bonding. This is not true. Carbon doesn't participate in hydrogen bonding. Sphere of hydration is formed when polar molecules form hydrogen bonding with surrounding water molecules. This statement is true. Sphere of hydration will be formed, for example, you have got this as your phospholipid. This polar head will form hydrogen bonding with surrounding water molecules while the tails will go inside. Mass spectrometry can be used to identify the type and amount of fat in a sample. True or false? So this is going to be a false statement. That is because mass spectrometry is used to find out the mass and relative concentration. Just a mass and the relative concentration of a substance. How much of a substance, how much of fat you have, not really for identification. For mass and concentration. You already identified what your compound is, but you want to find out how much you have. All right. And then the next question, which of the following is a nucleoside analog that will be used in treatment of HIV using HAART therapy? Now for this one, Teno, I don't know how you are going to pronounce this, but this same one, and also lamivudine, these are going to be able to be used to treat, these are nucleoside analogs that can treat HIV with the HAART therapy. All right, the next one. With regard to water molecule, which of the following statements are true? The atoms of water molecule have same electrical charges. They do not have the same electrical charges. You know that oxygen is partially negative on both ends, while these guys, the hydrogens, are partially positive. Polarity of water molecule makes it an excellent solvent. This is true. Negative charged atoms will be attracted to the positively charged oxygen. Now, oxygen is not positively charged. In water molecule, the hydrogen and in water molecule, the hydrogen and oxygen atoms create a symmetric molecule. Okay, this statement is not true. That is because water is not symmetric. It's actually an asymmetric molecule. So the best option there, the polarity of water molecule, makes it an excellent solvent. And then the next question, which of the following is not a pyrimidine? What is not a pyrimidine means that it is a purine, and you can use pure as gold. Pure as gold. Purines are adenine and guanosine. Adenine and guanosine. So which of the following is not a pyrimidine? It should be either adenine or guanosine. That's not a pyridine. So that is a D there. Okay. So that is the answer, which is a purine, I mean. Amylose is a linear chain of glucose molecule joined by alpha 1, 4 glycosidic bonds. True or false? So this statement is true. Amylose is joined by alpha 1, 4 glycosidic bonds. It is linear. Amylopectin is branched. Which of the following statements are true? During dissociation, atoms can either gain during dissociation, atoms can either gain or lose electrons. Okay, so we need to know the difference between ionization and dissociation. To ionize is where a compound forms an ion by losing or gaining electrons. Say, for example, sodium loses an electron to become sodium ion. It has ionized. But this association is where something gets to break to form ions while dissolved in water. For example, I've got the salt sodium chloride. After it dissolves in water, it can form sodium ions and chloride ions. So that is this association. It has to do with being dissolved in water. So during this association, atoms can either gain or lose electrons. Is this true? No. During ionization, ionic compound split. Is that true? Immediately they mention ionic compound split. That is not ionization, it's actually dissociation. Water molecule can be dissociated into hydronium ion and hydroxide ion. Is that true? This is true. That is because if you have got water, this one can dissociate into S3O plus and also H minus. Actually, two water molecules here, they're supposed to react, where one water molecule is going to donate a proton so that it forms H3O plus. Addition of acid decreases the concentration of hydronium. Is that true? Now, hydronium is this. When you add an acid, hydronium is supposed to increase, not decrease. So that is not true. So the best option there is C. You are in the lab and asked to change the net proton charge so that the protein can be denatured. Which of the following would you do? Add urea. Okay, so on denaturing of proteins, proteins can be denatured. Only the secondary, tertiary, and quaternary protein structures can be denatured. The primary, structure can't be denatured. Okay, so you can denature proteins by doing three things. You can alter the pH. Now, if you alter the pH, how does pH denature the protein? It denatures the protein by altering the net protein charge. That is how pH will be able to destroy a protein. And then temperature can also destroy or denature a protein. How does it do that? It gets to disrupt hydrogen bonds. And then you have got also things like alcohol, urea, and detergent soaps. This can also destroy proteins, but they do this by disrupting hydrogen bonds. Not hydrogen bonds, but hydrophobic bonds. So this is why they do. So for this one, we want to do what? To change the net protein charge. So what should we do if we want to change the net protein charge? We need to alter the pH. And which one is that? That is D. Of the HIV key protein, which one is responsible for regulation of viral mRNA expression? About regulation of viral RNA expression, we need to know that is REV. REV is involved in regulation of viral mRNA expression. Which of the following best describes a proton motive force? Proton motive force is where you have got, let's say, positive charges at one end and then there are negative charges on the other end such that electrons are able to move like that, generating energy. A pH gradient created by energy from electron movement. This is true, a pH gradient. What happens is that you are going to have more hydrogens at one end and then fewer hydrogen on the other end. That is what creates a pH gradient. And this is because of electron movement in the electron transport chain. An electron gradient created in the mitochondria is not electron gradient really. The movement of protons from one region of the mitochondria to another. This is true. Okay, when protons, hydrogen move from one region of the mitochondria to another, actually they move from the matrix of the mitochondria to the intermembranous space there. The movement of protons and electrons to generate carbon dioxide and water, that is necessarily not proton motive force. Which of the following best describes the flow of electrons during redox reactions? The flow of electrons during redox reactions. Electrons move randomly through the electron carriers. Is that true? Now, electrons don't move randomly in electron carriers. Electrons move to nearby electron carriers in the electron transport chain. This is true. Electrons will move from complex 1 to complex 2 to complex 3 and just like that. Electrons move through electron carriers that are arranged in close proximity and with an increasing affinity for electrons. This statement is also true. Electrons can only move from complex 1 to complex 2 because complex 2 has got a higher affinity for electrons. Electrons move towards electron carriers that are arranged in close proximity with reducing affinity. That's not true. When equilibrium constant is negative, a reaction will move in the forward direction, favoring formation of product. True or false? Let me just show this. Let's say you have got A going to B, right? When equilibrium constant is negative. Now, about equilibrium constants, when the equilibrium constant is less than 1, meaning that it is negative, this will mean that delta G is actually going to be positive. And this reaction will move in the backward direction. Every time you see delta G is positive, it means that the reaction is not spontaneous. So the reaction will flow in the backward direction. When K is equal to 1, delta G is going to be equal to 0. And this reaction is at equilibrium, not moving in any direction. When K is greater than 1, delta G is going to be negative. This reaction is spontaneous. So this reaction is going to move in a forward direction. So when equilibrium constant is negative, a reaction will move in a forward direction. Okay, as we can see here when equilibrium constant is negative. Okay, which is this one. Reaction will move in the forward direction, favoring formation of product. True or false? So this statement is false. It is just opposite of what we have said here. Which of the following monosaccharides contain an aldehyde group? So they should have the carbonyl group at the end of the chain. Galactose has got the carbonyl at the end of the chain. Ribulose is a ketone, unless ribose. Mostly if you see rulose, just know that those are going to be ketones. For example, ribulose, xylulose, unless if it was xylose, not xylulose. Mostly if you see a U and then LO, those have got ketones. Fructose also can see a U there, that's a ketone. So the answer is the first one. Felling's test for qualitative analysis of sugar works on the principle that, now Felling's tests are used to identify aldehydes. And for Felling's test, what happens is that the carbonyl group of a reducing aldose sugar is going to react with a reagent to be able to produce a color change. This is true. So the carbonyl that is present in the sugar will react with the reagent and then you're going to be able to see a color change. The hydroxyl group of a reducing sugar can be oxidized. That's not the principle. The presence of a reducing sugar can can oxidize copper 1 to copper 2. This is actually the direct opposite. It's supposed to be copper 2 to copper 1. So this is not true. Reducing sugar will be reduced to N-diols. That's not true. Glycoprotein are formed when oligosaccharide are bound to protein. True or false? So we need to understand the difference between glycoprotein and proteoglycan. From the word glycoprotein, of course, this statement is actually true. It's formed between oligosaccharide and the protein. But if it was proteoglycan, proteoglycan is also having a protein and a carbohydrate. But the specific carbohydrate that you are going to have is a glycosaminoglycan. But here the specific carbohydrate you have in a glycoprotein is oligopolysaccharide or oligosaccharide, sorry. And then cholera enterotoxin activates adenyl cyclase, resulting in an increase of which nucleotide? So the cholera enterotoxin increases cyclic adenosine monophosphate, and this will promote the entry of water into the lumen of the intestines, and that causes diarrhea. All right, so that is it. The signaling molecule prostaglandin E is involved in the following signaling pathways. Now, prostaglandin E is involved in the contraction of smooth muscles. When the smooth muscles in blood vessels contract, that is known as vasoconstriction, not vasodilation, because this is relaxation. Muscle construction. So this statement has just changed the meaning. It's not muscle construction, but muscle contraction. So if it was a mistake, but I don't believe it was a mistake, so that is not true. Constriction of airways. This is going to be true because airways are made up of smooth muscles. Increased platelet recruitment, really not. Bioenergetics describe the simultaneous anabolic and catabolic processes in a living organism. True or false? So this is going to be true. Bioenergetics will talk about both what make how energy is made and how energy is broken down. Zidovudine, one of the nucleoside analogs of HIV treatment, becomes insufficient due to. So if you just see where there's looking as though it's money, K70R, just know that's the answer. So it is a deficiency of these K7N and K70R amino acid mutations. So this is it. Positive value suggests that electrons move from the test cell to the hydrogen cell. True or false? This statement is false. So if you have got this as your hydrogen cell, this is your test cell, and then you're having a voltmeter there. If it shows a positive value, it means that electrons are moving from the hydrogen to the test. Okay, not from the test to the hydrogen. Which of the following statement is true? Serine and threonine participate in enzymatic reactions due to their thiol group. This is true. Amino acids are colorless because they do not absorb UV light. This is false. They are colorless because they don't absorb visible light. Catecholamines are derived from tyrosine. This is true. Proteins absorb at 260 nanometers because of the aromatic amino acids. This is not true. Because of the aromatic amino acid, they absorb at 280, not 260. Which of the following methods would be the most suitable to distinguish between fats that have the same number of carbon atoms but different levels of saturation? So for this one, we can use a high pressure liquid chromatograph to be able to differentiate. Which of the following statements is false? Aspartic acid is an uncharged derivative of asparagine. This statement is false, right? Because aspartic acid is an uncharged derivative not of asparagine but of aspartate, just like glutamic acid is an uncharged derivative of glutamate. And then arginine is conditionally essential. This statement is true, so it can't be the answer. Alpha-mantine is a toxin found in some mushrooms. That is true. It is also found in antibiotics. That is also true. So the only false statement is that one. Okay. Samsung Erickson is in the lab trying to target peptide bonds. Which of the following tests would she use? Ninhydrin reagent. This one does not target peptide bonds, just like the Eddington reagent. These target the carbonyl and the amino groups. So also the Sanger's reagent doesn't target the peptide bonds. What targets peptide bonds is the biorate. Reduction of aldehyde groups produce gluconic acid. True or false? Now this is false because when you reduce aldehyde groups, what you actually produce are aldetors, and an example of an aldehyde is going to be sorbitol. Now, when you get to oxidize carbohydrates as C1, you are going to produce aldonic acid. And an example of aldonic acid is gluconic acid. If you oxidize at C6, you produce uronic acids For example, glucuronic acid. These are oxidations. If you oxidize both C1 and C6, you are going to produce aldaric acid. Example, glucaric acid. But reduction produces alditose. An example is sorbitol. Nuclease is a class 3 enzyme. Which of the following is true? Which of the following is true? It does not require a coenzyme. True or false? This is true. Class 3 and class 4 don't require a coenzyme. It does require no. Catalyzes reaction that transfers groups? No. Catalyzes reaction that transfers electrons? No. Okay. So the best option there is option number 1. Actually, class 3 enzyme, you can use this mnemonic to represent the classes: class 1, class 2, class 3, class 4, class 5, class 6. Class 3 enzyme, these are hydrolyses, are involved in hydrolysis reactions. That's why the other options are incorrect. Methotrexate is an anti-cancer drug that binds to the same site as tetrahydrofolate, a coenzyme for the enzyme dihydrofolate reductase. This type of inhibition is referred to as. So this type of inhibition, because methotrexate binds to the same site where the substrate binds, that is competitive inhibition, but it can be reversed, so it is competitive reversible. Okay, that's the answer there. The carbonic acid bicarbonate system is closely associated with the respiratory system. This option, this is true because the carbonic acid bicarbonate system actually produces carbon dioxide. And this carbon dioxide is going to be breathed out because we are going to face the carbonic acid being decomposed into hydrogen and the bicarbonate. And this same reaction can also get to produce, you can also have water reacting with carbon dioxide getting to produce bicarbonate. But this same bicarbonate can also go in a reverse reaction using the enzyme carbonic anhydrase, producing carbon dioxide, and that is going to be expelled. Which of the following best describes the feature of fats that make them an ideal energy storage macromolecule? They don't interact with water, therefore they don't affect cellular osmolarity. This is true. This is why they are good for storage. They produce a lot of energy. This is an importance not for storage but for production of energy. They are not highly oxidized. They are long chain hydrocarbons. This is important not for storage but for production of energy. Unsaturated fatty acids refer to unsaturated fatty acids. These are fatty acids that are going to have a double bond somewhere. Fatty acids with a high melting point, they have got a low melting point with a double bond. This is true. Okay, fatty acids without, this is not true. Fatty acids that appear solids, these are actually liquids because of a double bond. Which of the following statement is true? Now I want to talk about the Michaelis-Menten equation and the double reciprocal graph. You are going to be able to see this graph like this for the Michaelis-Menten kinetics. And this one, 1 over VO is equal to, what is the equation again? Okay, that is 1 over VO is equal to Km divided by Vmax multiplied by 1 over the concentration of the substrate plus 1 over Vmax. So here this is in the form Y is equal to Mx plus C. So we need to know the interpretation. X-intercept is represented by negative 1 over Km. So this one is saying given by Vmax, that's not true. Y-axis intercept is given by Km. You can see the Y-intercept is given by 1 over Vmax, that's not true. High Km indicates weak binding, this is true. Allosteric enzyme partially obey the Michaelis-Menten kinetics, this is not true. Allosteric enzymes do not obey the Michaelis-Menten kinetics. Vaccine quarantine is a medical student trying to separate a mixture of protein based on size. Which method would you advise them to use? So separating proteins based on size, you can use gel filtration. Affinity chromatography is not based on size, right? But it's based on affinity. An ion exchanger is based on charge. All right, so that is it. You are in the lab and asked to add an amino acid that can act as either a base or an acid at the pH of 7. Which one would you choose? So an amino acid that acts both as an acid and a base at neutral pH is histidine. So that is D. Which of the following processes need to occur for a nucleoside analog to function? Phosphorylation has to occur for a nucleoside analog to function. Which of the following is correct? Fatty acids have higher melting temperatures compared to unsaturated fatty acids because of interchain interaction. This is not true, unless maybe they said saturated fatty acid, but they've just said fatty acids. Of course, saturated ones have got a higher melting point than unsaturated ones. Saturated fatty acids do not have free rotation about their carbon to carbon bond. This is not true. They have got free rotation. What doesn't have free rotation is unsaturation, because they have got a double bond. Saturated and unsaturated fatty acids with the same number of carbon atoms, you have comparable melting points. That's not true. The melting point of saturated ones are higher. Unsaturated fatty acids have chains spaced far apart, resulting in a lower melting point. This is true, because if you look at the way these are the unsaturated ones, they are going to have a kink because of a double bond. And so the next chain will have a bit of distance away from that one. So that lowers the melting point. A lab technician is attempting to cleave peptide bonds and peptides and triglycerides. What enzyme would you advise them to use? Peptides can be cleaved by substituents and triglycerides can be cleaved by glycosidase. So the best option that we can pick here, substitutes. Oh, this is actually triglyceride. Glycosidase cleaves carbohydrates. This is triglyceride. This is a lipid, so it's lipase. So the best one will be this one. The larger the value of the acid dissociation constant, the stronger the acid. True or false? Acid dissociation constant is Ka, and the larger the Ka, the stronger the acid. This is true. But if it was pKa, the lower the pKa, the stronger the acid.